Hi, my name is Julian, and I'm a rising junior at Brown University, and I'm studying biology and data fluency. This summer, I've had the opportunity to participate in the NIH SenNet CUSP internship program at the Dixit Laboratory, where I examine P16 and P21 as senescence biomarkers in various immune cells of wild-type B6 mice. The Dixit Lab is based in Yale University, which has two TMCs that map senescent cells in lymphoid organs. I was in the murine TMC, and Mira, another CUSP student, was in the human TMC. But my lab specifically studies immunosenescence, immunometabolism, and aging, focusing on the relationship between immune and metabolic systems in murine models. So for some context, senescence is permanent cell cycle arrest induced by stress and damage, characterized by the lack of proliferation and upregulation of anti-apoptotic pathways. Early in life, it is important for normal development and morphogenesis, and later in life, it's essential to tissue repair and inhibition of cancer outgrowth, which many applications clinically are seen today. Senescent cells are found to have accumulated with age, and it's likely due to the fact that the immune system's capacity to eliminate senescent cells becomes dysfunctional with age and disease. Senescent cells are also very heterogeneous across cell types, tissues, and timescales, and there are an unknown number of phenotypes. So the study of senescent cells remains an ongoing area of research. There are 9 principal hallmarks of senescence, but the one I've focused on is cell cycle arrest, which is characterized by the upregulation of P16, P21, and P53. P16 and P21 are both CDK inhibitors, which inhibit E2F transcription factors and downregulate MKI67 proliferation markers, leading to cell cycle arrest. They are found to accumulate in senescent cells, and there isn't one exclusive universal marker for senescence, so P16 and P21 are widely studied. The two lymphoid organs I focused on were the spleen and thymus. In the spleen, there are two regions, the red and white pulp regions, separated by a marginal zone. In the red pulp region, there are splenic cords and sinusoids for filtration of the blood. And the white pulp contains lymphatic tissue with germinal centers for humoral response development. When senescence occurs in the spleen, the senescent environment impairs functionality of other spleen cells, such as T cell recruitment, which can evidently lead to immune dysfunction. And as for the thymus, there are also two general regions, the outer cortex area and the inner medulla area. And the outer cortex area is known to have a higher density of thymocytes, macrophages, and epithelial cells compared to the medulla. Aging and senescence in the thymus causes thymic involution, which leads to immunosenescence and inflammaging. So there's significant functional decline as cells get replaced by fibroblasts, adipocytes, and senescent cells. So there are many different implications of senescence research, but xenotherapy is one that is especially pertinent to our work. The therapeutic clearance of senescent cells could help restore thymic and splenic function, specifically through drugs like senolytics, which selectively eliminate senescent cells that have detrimental pro-tumorigenic effects. Especially in anti-cancer therapy, senolytics are used to target P16 and P21 as regulators of stemness. So for my project, I focused on determining the tissue-specific distribution and cell type-specific prevalence of senescence in immune cells. My experiments utilized immunofluorescence staining for P16 and P21 biomarkers in splenic and thymic immune cells of young and aged wild-type B6 mice. The young mice were around 3 months old and the aged were around 25 months old. I studied senescence in 3 different immune cells, the epithelial cells, macrophages, and fibroblasts, as well as compared them with proliferative cells. And on the right, I have a little chart of all the antibodies used for each of the biomarkers. I referenced various IHC and IF protocols, but my general method was to first dissect the spleen and thymus from the young and aged mice, Then I froze the tissues in OCT blocks. Then I serially sectioned the tissues with a cryostat machine. And then I mounted them onto the slides and stained them with their respective antibodies for IF staining. And then finally, I imaged the slides with a fluorescence microscope, specifically the Keyence fluorescence microscope. So as an example, these are some of the images I took for a young spleen sample stained for cell nuclei with DAPI in blue, the P21 senescence marker in green, and then the macrophages in F480 in the red. And in the overlaid image, we can see the yellow cells that show the colocalization of P21 and F480, indicating that there is presence of P21 in the splenic macrophages. And with an aged spleen sample, we can see that there's a lot more colocalization of P21 and macrophage cells, again seen in the overlaid image here. Repeating the imaging with both P16 and P21 staining, we can compare them side by side and see that P16 and P21 are prevalent in splenic macrophages, and more so in the aged compared to the young mice, and also more so in the inner darker white pulp areas than the outer lighter red pulp regions. Looking at the thymus, we see a similar trend. P16 and P21 are prevalent in macrophages and more so in the aged compared to the young mice. We also see that they are more densely clustered in the outer cortex area as opposed to the inner medulla area of the thymus. Now looking at epithelial cells, we again see colocalization of P16 and P21 in the spleen, with a higher density in the white pulp region than the red pulp region, and again a higher density in the aged mice compared to the young mice. For epithelial cells in the thymus, there is a similar distribution of colocalized cells. We see that P16 and P21 are both prevalent in the thymic epithelial cells, and again more so in the aged samples than the young samples, and more concentrated in the cortex outer area than the inner medulla area. Regarding fibroblasts, we see that P16 and P21 are once more prevalent in the spleen, with a higher concentration in the center white pulp region as opposed to the outer red pulp region. And they are more concentrated in the aged mice as well compared to the young. Though in the thymus, we actually found that there was virtually no colocalization visible, leading us to believe that senescence may not occur in thymic fibroblasts. We have yet to look into the reasons behind that, but that is something to consider in the future. In addition to having unstained negative control slides, we also stained for proliferative cells to confirm whether P16 and P21 antibodies were working properly. And for the most part, we see that there's not much colocalization in the spleen of young mice, but we do see a little bit in the aged spleen, especially with the P16 old spleen sample. Since proliferative cells would not be exhibiting senescence by definition, this abnormal staining gives us inconclusive results. So we can't confirm with full confidence the specificity and efficacy of the P16 and P21 antibodies that we used in the stainings. To summarize the results, we found that both biomarkers were more prevalent in age than young mice, and P16 and P21 were more prevalent in the cortex area compared to the medulla and the thymus, and in the spleen more prevalent in the white pulp regions compared to the red pulp regions. And both biomarkers were prevalent in all three immune cells examined in the spleen. However, in the thymus, they were present in all but the fibroblasts. Finally, our confirmation of the senescent cell stainings was not the most successful, so that is something to address and examine in the future. Some future directions to take this research could be to repeat the stainings with other immune cells such as Th and TUB3, which would stain nerve cells. And I only imaged with a Keyence fluorescence microscope but imaging with a higher resolution, like confocal microscopes, could result in higher quality images and analysis. Finally, my lab was predominantly a wet lab environment, but having IHC and H&E stained tissues analyzed computationally, such as through single cell and spatial omics analysis, would enable more detailed examination of senescence within the target organs and cells. We even sent some samples to the fan lab where the other student Mia was working at, and they were able to work on the codex and DeepSeek analyses. To conclude, I wanted to give thanks to the Dixit Lab and the NIH members who enabled this entire research opportunity. I'm grateful for the mentorship of my PI, Dr. Dixit, and lab mentor Uni, as well as everyone else in the lab I got to work alongside. I also want to thank the NIH Common Fund and its donors for supporting the CUSP program. And of course, thank you to the NIH SenNet members and guest speakers, the other CUSP interns, and the CUSP organizers, especially Kay and Elizabeth, for shaping the entire SenNet CUSP experience. This program has empowered my growth on the professional, academic, and personal level, and I look forward to harnessing and further developing the knowledge gained in my future career as a scientist. Thank you.